Starting off our second unit, we need to learn some things about carbon and about biochemistry. Now, you've already learned a lot of these things before in your first biology course. That doesn't mean you remember it all, so I'm going to review over the basic things about carbon chemistry and add some stuff that you may not have learned about in either chemistry or biology. And then we're going to talk about in this first set, we're going to talk about carbohydrates and lipids as well as the basic chemistry stuff, and then we'll talk about proteins and nucleic acids. So first off, to begin with, where organic molecules came from, there's a lot of controversy and a lot of people think different things, and nobody really knows because no one was here when the Earth was formed. But there have been several theories about where organic molecules could have come from. And of course, nowadays a lot of people are thinking maybe there were comets that carried organic molecules from other places, or meteorites even. But the classic experiment that was done way back in the early 1950s was Stanley Miller's classic experiment that demonstrated that organic compounds could be synthesized from abiotic origins. And there are other experiments since then. The ideas about what the compounds probably were in the early atmosphere and oceans have changed since Miller's time, but still there have been modern experiments that have followed the same line of thinking that support that idea that that organic compounds could have been from abiotic sources rather than from extraterrestrial sources. So we don't really know right now, and there are lots of different theories about it, but we're just going to talk about these experiments because this is kind of a classic biology thing. So the Miller-Urey experiment that was done in like 1951 had a chamber like this. They had a sea water, basically with water in it, and that was just all that was there. They exposed it to heat to boil the water. The water vapor then came in contact in the second chamber with chemicals that at the time were considered to have been some of the compounds that could have been present in the early atmosphere, and that included methane gas and ammonia and hydrogen gas. Nowadays we have different ideas about that, but that's what they were thinking at the time. They exposed the mixture of gases here to electrodes to simulate lightning, and then the gases then, as they cooled, went through a condenser to cool them down to make a liquid water again, and they let it run for a period of time. I don't recall the exact period of time they let it run, but it was probably like a week or two at least. And then they bled off some of this fluid that had been condensed from this atmospheric mixture with the lightning, and what they did was they analyzed this solution chemically to see what things were there, and they found that there were some primitive amino acids and other kinds of organic compounds that could have been formed. So definitely organic compounds could have been synthesized from these kind of origins. Okay, nowadays they have different ideas about what the compounds probably were in the atmosphere. Nowadays they think more like carbon dioxide and nitrogen gas and some of those kind of things, but similar experiments with the current thinking about the atmosphere have resulted in similar results. So the idea for the experiment that they came up with is really a pretty good one. So we're not going to worry too much about where the compounds came from. We're going to worry about what we're going to do with them. And the thing that you need to realize about carbon, if you haven't realized this before, or just as a reminder, that carbon can form covalent bonds with a lot of different atoms, okay, since it has those 4 valence electrons. And this lets you have the opportunity to make large complex molecules, okay. Carbon can bond with itself easily. It can bond with other things as well. When you have multiple carbons, the carbon basically is bonded to 4 other atoms with a tetrahedral shape. But when you join 2 carbon atoms together by a double bond, then that's going to change the position of the other things that are bonded to carbon. And what you'll find is the atoms that are joined there are now in the same plane as the carbon rather than being in a more three-dimensional shape. So here we have some of the basic carbon compounds. Okay, we have methane here. In a structural formula we show just the hydrogens evenly spaced there, but the ball and stick model gives you a better idea of the three-dimensional structure of the molecule. It's a tetrahedral shape here, and the space filling model of course gives you an idea about where the electrons are. Ethane, we've got 2 carbons and 6 hydrogens. And again, a more complex three-dimensional structure here in the ball and stick model, and the space filling model as well. And then ethylene, same thing here, except we've got the carbons double bonded to each other, which means that you have fewer hydrogens, C2H4. And so you'll see now, instead of this three-dimensional shape, now we go back to a two-dimensional shape, which is a lot different because of that double bond. So what you need to realize about carbon is that you have a lot of diversity in molecules that can be produced from a carbon skeleton, and the carbon skeletons can vary in different ways. There are different kinds of shapes that can form from the carbon skeleton, and the carbon chains can vary in length and shape, and so we'll see some examples of that in the coming slides. Here we have 4 different ways that carbon skeletons can vary. First of all, by length. Okay, all you got to do is just add more carbons to the chain, and that gives you a longer and longer chain: ethane, butane, propane, butane, pentane, hexane, and so forth. Okay, you can have instead of a long chain, you can have branches, side branches. And so here you have 2 molecules that have the same molecular formula, which is C4H10, but here we have a branching structure that is different in its shape and different in its reactions as well. So we have, instead of butane, we have isobutane or 2-methylpropane. Propane, because it's based on this 3-carbon chain here with one side chain off to the side. Then you can change the position of a double bond. If you have double bonds in your basic chain, you can change the position. Here's one butene that has the double bond between the first and second carbon, and then you have 2-butene that has the double bond between the second and third carbon. Again, they have the same molecular formulas, but their structural formulas are different and they'll react differently. And then the 4th way that a carbon skeleton can vary is the presence of a ring structure. Oftentimes there are gases that form this way, but oftentimes in solution, carbon compounds will form into this ring structure like this. So here we have cyclohexane. Again, it's hexane, but it's got a little bit different molecular structure because you've got this ring structure, and that's going to cut down on the number of hydrogens as well. And then benzene, where you have alternating double bonds between those carbons, a very stable structure. This is one of the chemicals that used to be used a lot for solvents and things like that, but it's actually found to be pretty harmful because of its stability. It doesn't break down very easily. So in addition to the basic carbon chain and the variations that we can have there are also some chemical groups that are components of these organic molecules that are often found in various kinds of chemical reactions, and these are things that are more commonly found in living things. The ones that we're going to be concerned with are they're the most common functional groups in living things. And so we're going to talk about the number of those and arrangement of them and so forth that give these molecules their properties. The 7 functional groups that are most important in this chemistry, and this is really important: the hydroxyl group, the carbonyl group, carboxyl group, the amino group, sulfhydryl group, the phosphate group, and the methyl group. You may or may not have talked about some of these in chemistry. It doesn't really matter because we're talking about them in biology, and it is important for you to know them. You need to know these 7. You need to know their you need to know their molecular formulas. You need to be able to recognize them in various kinds of structural formulas and molecular formulas of various carbon compounds that we'll be talking about. Really, really important. We're going to talk about each one in turn. So first of all, the hydroxyl group. Hydroxyl is OH. Okay, it's the hydroxide ion, basically. And hydroxyl groups are found in alcohols, okay? Sometimes you'll see it written OH, sometimes it's HO, depends on which end of the molecule it's shown on. But alcohols generally have a name ending in OL, like propanol, isopropanol, ethanol, and so forth. The OL part means the hydroxyl group, and that's OH. Then we have the carbonyl group. A carbonyl group is a carbon double bonded to an oxygen. This makes a ketone if the carbonyl group is within the carbon skeleton, like in the middle of it somewhere. If the carbonyl group is at the end of the carbon skeleton, it's called an aldehyde. And probably the one that you probably have heard of more than anything else is formaldehyde, okay, which is a very simple compound with one carbonyl group on it. But you need to be able to recognize a carbonyl group as separate from a carboxyl group, okay, which we're going to talk about next. The carbonyl group is just a carbon with a double bonded O, and the carbon is attached in the carbon chain, and there aren't any other hydrogens or anything attached to that carbon. Our carboxyl group is usually found at the end of a molecule because you've got to have both of the double bonded oxygen of the carbonyl group and the hydroxyl group attached to the same carbon. And so you will usually see this at the end of a carbon chain. These are the carboxylic acids, or the organic acids. So anything that is an acid that is an organic molecule would have a carboxyl group on it. That's the COOH, or the COO group if you want to call it that. But again, it's a combination of the carbonyl group and the hydroxyl group, and it is found on the end of a molecule. Carbon, remember, only has 4 places to bond, so it's got 2 of the bonds taken up with the oxygen, 1 with the hydroxyl group, and then the 4th bond is going to be attached to the carbon chain. The 4th one is the amino group. Amino group doesn't have any oxygens in it, but it has nitrogen and hydrogen. It's an NH2 group. These form the amines. And so now you see why we talk about amino acids, because the amino acids will have both an amino group and a carboxyl group, so that's why they're called amino acids. The amino group is usually basic, believe it or not, and so an amino acid has an acid on one end, and on the other end it has a basic structure, and oftentimes those side chains on amino acids change the structure of it altogether, or the pH of it altogether. The 5th is the self-hydryl group. Self-hydryl is the sulfur with a hydrogen, and these are the thiols. So anything that's got thiol in it is going to have that self-hydryl group. The only thing I can think of off the top of my head that has that in it is an old compound that they used to use to put on cuts and things. It's got iodine in it, and it was called merthiolate, and it's got that thiol in it, and it's also got iodine in it as well. The phosphate group is another one that you should be familiar with. Phosphate is PO4. You've got the phosphorus that's attached to an oxygen, and this oxygen here is going to be attached to the carbon chain. You've got 1 double bonded oxygen, which is the carbonyl group, remember, and then you've got 2 oxygens here. Normally this is going to be an organic phosphate, and normally these oxygens are going to be probably have a hydrogen attached to them as well, but they're going to form a something that is definitely got a polarity to it. And then you've got the methyl group. The methyl group is basically a methane group on the end of a carbon chain, and so that's going to be a methylated compound when you have the CH3 methyl group on there. So 7 functional groups that you need to be very familiar with: hydroxyl, carbonyl, carboxyl, amino, sulfhydryl, phosphate, and methyl groups. You need to know those backwards and forwards and recognize them in structural formulas as well as molecular formulas. Now, let's talk about macromolecules in general. Okay, macromolecules are polymers, and they're built from monomers. The monomer is the small piece, the one Lego brick, and the polymer is the long chain of all of those put together. Okay, it's a long molecule made of a bunch of the similar type things. They're not necessarily all exactly like. They can be, depending on what the molecule is. The small ones are called the monomers, okay, and the big ones are called the polymers. 3 of the 4 classes of organic molecules are polymers: carbohydrates, proteins, and nucleic acids. The 4th class is the lipids, and lipids are not true polymers, although they are made of a couple of smaller molecules. But we'll talk about why that is when we talk about lipids. When you put together monomers to make a polymer, you undergo a reaction called dehydration. Now remember back to Biology 1, or pre-AP biology, we talked about dehydration means the removal of water. And so what's going to happen is you're going to combine these 2 molecules together. You're going to remove an oxygen from one and a hydroxyl from the other, and the hydrogen and the hydroxyl are going to go together to make water, and they're going to leave together a place where the 2 molecules can be joined together. So the dehydration reaction, or dehydration synthesis, is how you're going to put these monomers together. You're losing a water molecule for every bond that forms between monomers. When you break down polymers to monomers, the process is called hydrolysis. Remember, hydro means water, lysis means breaking down. It's essentially the reverse of the dehydration reaction. Hydrolysis is the process that your body uses in the digestive system to break down the foods that you eat into the component molecules that will be used by your body to produce other compounds. Okay, so hydrolysis basically is digestion. That's kind of what's happening in digestion. It's breaking down the big protein into smaller amino acids, the big carbohydrates into smaller monosaccharides, and so forth. So these two reactions are basically the reverse of each other. It is very important that you understand how they work. Here we have dehydration synthesizing a polymer. So we have two short polymers that are not linked together. One has a hydrogen on one end, the other one has a hydroxide on this end. So what we're going to do is we're going to remove the hydrogen and the hydroxide here, combine those two to make water, and that will leave an open bonding site here on this carbon and on this carbon where they can join together. So dehydration is going to remove the water molecule, forming a new bond and a longer polymer in the process. This can happen multiple times. Usually this is catalyzed by an enzyme, and we'll talk more about enzymes later on. But usually this is catalyzed by an enzyme, and the enzyme that catalyzes this reaction is specific to the reaction for whatever it is that you're adding together. Hydrolysis, the opposite. Okay, we're going to use it to break down a polymer. Again, you're going to have an enzyme where this molecule is going to fit, is going to put stress on this bond between these two carbons, causing it to break, and you've got the hydrogen and the hydroxyl group there ready to jump in and take their place to break down our longer polymer into two shorter monomers. And I said we were going to talk about carbohydrates and lipids in this particular segment, but I'm looking at the time and it's almost 17 minutes, so we're going to stop here and I'll make another recording specifically about carbohydrates and lipids.